Welcome, dear students. Today we have to discuss about deterioration of crop varieties. This is very important topic in seed technology, you see. Of course, crop varieties are very important. But if you are unable to provide the quality seed of those crop varieties, the purpose of developing of new crop varieties is defeated. So you see what we plant breeders or seed technologists are doing to preserve the genetic purity of the crop varieties. We maintain the crop varieties from its deterioration. So what we used to do, why farmers are asked to replace their seed if it is self-pollinated, every 2-3 years they have to replace, they have to take new seed from us, from the breeders or from the seed technologist. What we are doing to preserve to maintain the purity of the crop varieties. So for that we must understand that what are the causes of deterioration of crop varieties. This is very, very important in seed technology. If we try to understand simply these important things, we are going to solve the problems. So please, we can discuss one by one the causes of deterioration of crop varieties. The first is developmental variation. If we try to understand this, while producing the seed of quality seed of crop varieties, we can address this problem. What is this? What does it mean? Developmental variation means variation is coming because of the developmental stages. What is that? Means if you are producing the seed of a particular variety in those areas where it is not developed, for which area it has not been developed, so you are producing the seed in other areas, in other agroclimatic zone, in other environment, in other climatic conditions. In due course of time, you will be finding there are some differences coming in your variety because of the environmental changes and because of the adaptation to those environment, to this variety which is not developed for those areas. So if you are producing generation after generation those seeds of those varieties in those environment where it is not developed, it is not developed for, or for it has not been released for. So because of that, they get adapted with the changes coming because of the differences in the environment, and they are adjusting to those environment, and after few generation, in due course of time, they get adjusted to that particular environment because of the variations and adaptation. So your variety gets changes, genetic changes in that. So there will be genetic differences if you are producing generation after generation in other environment. So you have to keep in your mind that if you wish to preserve your variety, not to deteriorate your variety, genetically you have to produce the seed in the areas for which the varieties have been released. So this is all about developmental variation. We have to keep in our mind. This is very, very important. Then come to mechanical mixture. Of course, we have to be very, very cautious while producing the seeds. What happened? You see, you are going to grow seed crop. You have to see the field as per the rule, whether that field is eligible for producing that particular variety or not. Say for example, if it is rice, you are going to produce the seed of a particular variety, maybe HUR 105, and last year the rice seed was produced in that particular field, the variety was another variety, maybe Swarna. Last year you have grown Swarna, this year you are going to grow this HUR 105. So what do you think? So please don't do that. You keep in your mind that if you have grown previous year the rice, the Swarna, and this year you are going to grow HUR 105. So if you see in the season, lot of volunteer plants, this year you are growing HUR 105, lot of plants are of previous year Swarna are coming up. So there are ways also. If you are not having other field for this variety, you are bound to do that. If you are having, so far so good, that is best, change it. But if you are not having, if you don't have scope, so there is another provision which will be dealt in another lecture. But try to see that there should not be mechanical mixture. This comes under mechanical mixture. This is one when you are selecting the field. Then next, when you are using the implements for sowing, seed drills and other things, you have to clean your seed drill properly. You must be assured that no other seed, even single seed, is available in your machine which you are using for sowing. So that point must be taken care of to avoid the mechanical mixture. Then again, what you are going to see when you are going to harvest. At the time of harvesting, also, if in nearby field you are growing another variety or other crop maybe, there may be mixture at the time of harvesting. You have to be very, very careful. And then if you are lifting your harvested material to threshing floor, there also you have to avoid this mechanical mixture. Then you are using threshers. There also you have to see that not a single seed is left other crop variety, other varieties in your threshing machine. Then if you are packing, at the packing time also in gunny bags, there should not be other seeds. Even in your seed drums, there should not be other seeds. in your processing machine, in your grading machine, at every stages of seed production and processing, you must be very, very sure that there should not be mechanical mixture. So your variety gets deteriorated because of these mechanical mixtures. So we have to keep in our mind that we must avoid these things. Then next, mutation. If you talk about mutation, the spontaneous mutation and induced mutation, you know that. The spontaneous mutation, you don't have any control. The natural mutation occurring, the mutation occurring in the nature, you know, is called as spontaneous mutation. If you talk about oligogenes, it varies from 10 to the power minus 6 to 10 to the power 4 to 10 to the power minus 6. Means 1 out of 10,000 plants or 1 out of 10 lakhs plant. This is the ratio of spontaneous mutation on oligogenes. This is occurring. So you are getting in 10,000 maybe 1 plant mutant. Maybe in 10 lakhs plant, 1 plant you are getting mutant. So these mutations are also creating hindrance, of course, in very negligible quantity. But in due course of time, those mutants may create problem. So you have to be vigilant for that also. And what you are, as soon as you are obtaining these mutants, they are looking off type. So as soon as you are observing, of course there are so many mutations, minor mutations, which cannot be identified. You cannot do that. You cannot identify and you cannot roll it out. So that much you have to observe in due course of time. You may get variations and genetic deterioration. So you have to keep in your mind this also, how can you solve it? So maybe if you are getting some variation, you go for roguing. So this is very important, this natural crossing, this is going on. Farmers are not able to see that. So as a seed producer, as a seed grower, you must understand this also. If it is self-pollinated crops, you know that up to 5% cross pollination is going on. So that is deteriorating your genetic purity. So that has to be addressed properly. And if it is often cross-pollinated crop, the outcrossing is increasing. More than 5% goes up to 30-35%. And if it is cross-pollinated, it is more than 40% outcrossing. So accordingly, you have to avoid this natural outcrossing in your seed field. Means you have to do something. For this, mainly the isolation distance is very, very important while producing the quality seeds. For avoiding, for checking this natural outcrossing, unwanted crossing because of the unwanted pollen grains. So this has to be addressed properly, and we are addressing these things in seed technology. That is why we are able to provide the quality seeds of the varieties. Then come to minor genetic variations. Of course, these variations at the time of release of the varieties or testing of the varieties, of course, we have taken proper generation, F6, F7 generation, F8 generation for going for the testing. and we have observed that, of course, there is homogeneity attained. Even then, there are some variations, minor variations, which could not be identified. The phenotypic variations, very minor, which could not be observed. But in due course of time, after release, it is seen. The minor genetic variations are seen. For some few genes, segregation is still going on for few genes, the minor genes. So that creates problem. For that also you have to go for purification of the varieties. Proper purification must be done in this situation. So next come to influence of pests and diseases. Of course, whatever new varieties you are developing, they are resistant and tolerant to different diseases and insect pests. But in due course of time, because of the mutation in pathogens, because of the occurrence of new races and pathotypes, your variety gets susceptibility to new pests and diseases. So your variety gets deteriorated. That becomes susceptible. If it is serious problem, then these varieties have to be stopped for crop production. So this has to be kept in mind, that this also comes in deteriorating your crop varieties. Then comes to techniques of plant breeders. Of course, we have to take care of this. Not the seed technologies here. Plant breeders have to take proper necessary actions for this, because if your technique is poor, if you are in hurry, and you have seen in your F5 generation that homogeneity is attained, then you have started your station trial and you have started sending your material to All India coordinated trial, and nobody could observe the segregation is still going on for many genes. So please, this is the problem of plant breeders. The seed technologies are not going to do anything. They have to just report. If it is released and it is coming in the seed chain, then seed technologists have to report this thing, that still this variety is having segregation. So please take it back and again try to attain the homogeneity, then you send it for release and sending it in seed production chain. So dear students, this is very important topic for seed technologists, that how your varieties are being deteriorated, and if you are understanding properly the causes of deterioration of your varieties, then of course you can do something wherever it is possible. You have seen already. So this is all about today's topic. I will be coming next with the next topic. Thanking you very much.